Good morning, everyone, and welcome to this year's first PGCTalks webinar series. To officially open the webinar, we would like to introduce our moderator, Ms. Shalette Monzon, PGC's Stakeholder Engagement Specialist.
Maayong adlaw sa ato mga estudyanteng halin sa Luzon, Visayas at Mindanao. And welcome to the PGC Talks 2021, a three-part webinar series spanning a range of topics on COVID-19, such as whole genome sequencing and its applications, concepts on a particular subset of data analytics called bioinformatics, and activities that the center is currently tasked to do. To formally start the webinar series, we would like to invite our Executive Director here at the Philippine Genome Center, Dr. Cynthia P. Saloma, to give her opening remarks.
So thank you, Shalette. Good morning, everyone, and welcome to today's episode of PGC Talks, a 3-week series of webinars that we at the Philippine Genome Center conducted or is conducting to communicate to you, the general public, as well as to our targeted stakeholders about any topic related to genomics and bioinformatics. So for today, we deem it timely to conduct this webinar series on the science and art of SARS-CoV-2 testing. We bring to you a simplified journey, a vicarious experience really, on how Rtpcr testing is done in our laboratories here at the UPPGC, from sample collection to Rt-Pcr testing, to interpretation of the CT values, to whole genome sequencing until the bioinformatics analysis part. Perhaps many of you are interested in how we detect variants among the swab samples submitted to us by the various laboratories and hospitals all over the country who form part of the Philippines SARS-CoV-2 Genomics Biosurveillance Network under the Department of Health. You will learn about this and many more as our speakers talk about these topics and their experiences at the UPPGC in a language that is simple, understandable, and targeted to our high school STEM students. It is our hope that through these efforts, we can inform as well as inspire you about how our local scientists, our molecular biologists, medical specialists, data modelers, and a multifaceted team use science to help in our coronavirus pandemic response. I encourage everyone, particularly the students in the audience, to listen closely and write your questions in the chat box and enjoy this learning experience. Good morning again, and let's begin PGC Talks. Thank you.
Thank you so much for that beautiful opening remarks, Dr. Saloma. We would also like to acknowledge the members of the PGC Executive Committee, as well as our colleagues from PGC facilities in the Visayas and Mindanao for joining us today. We will also be joined later on by our invited guest reactor all the way from Bacolod, Mr. Alain Siwayang, the Molecular Laboratory Section Head of the Corazon Locsin Montelibano Memorial Regional Hospital. Lastly, here are a few reminders we will be observing during the webinar. For our Q&A, selected questions will be posted on the screen after the lectures to be answered by the corresponding speaker or guest reactor. During the discussions, you can post your questions using the Q&A box below. There will also be a feedback form that will be presented in our chat room. The link will be provided in the chat room. For our live stream, this webinar is being live streamed currently using the Facebook page of the Philippine Genome Center. The link is on the screen. And for questions or immediate concerns regarding this webinar, you can email us using capacitydevelopment@pgc.up.edu.ph. Please see the link on the screen. Now for our first talk, let me introduce PGC's Clinical Genomics Laboratory Manager and current Clinical Health Officer who has been with us for the past 3 years, Dr. Mark Edsel CIS. He will be discussing the basic concepts of Rt-Pcr testing and how it is applied currently. Dr. IS, you may now share your screen with us.
Hello, good morning, everyone. I'm Dr. Aya. So I'll be talking about PCR, or what's known as the polymerase chain reaction. If I understand correctly, these are high school, no? So this will be I hope it's something that you've heard a lot about. It's been used as a trope in a lot of sci-fi and also mga crime-related shows. But the PCR is essentially it is the essential tools that we use in molecular biology, and it has extended its usage in the clinics in the form of molecular diagnostics. So I'll just be tackling the basics, the principles behind the polymerase chain reaction, so that you can get a greater appreciation of how it's actually utilized in the context of COVID-19. So this is the overall overview. We're going to talk about DNA, just a very short explanation so that you understand how it actually, how it informs the way that we perform the PCR, why we do this in order to study DNA, and how the polymerase chain reaction falls into that. How does it allow us to study DNA? And and then we'll go into how this technique has evolved into the way that we are currently using it in the pandemic. So to start off, if you've studied a bit in biology, you know that DNA, or deoxyribonucleic acid, is known as the blueprint of life. Now, it contains instructions in the form of genes, that we call it, that encodes for information that dictates how we grow, how we develop, and also how we reproduce as organisms in living biological systems. Now, you can think about it in the computer. You have information that's stored in the form of digital numbers like zeros and ones. But in the case of DNA, this code or these numbers come in the form of what we call bases, and they appear in base pairs. So there are 4 letters for them, A, T, C, and G, where A and T are known as pairs, and C and G are also paired together. So that's adenine, thymine, cytosine, and guanine. And they come in this sort of form that looks like a ladder that's been twisted at a certain angle. And you'll notice here that at the steps of the ladder, these are your base pairs. So G pairs with C and T pairs with A. And when you read it from the 5 to 3, meaning top to bottom, that sequence of letters, like in this example, you have G, T, A, C, that forms the basis of the instructions that would characterize genes, okay? And this is what forms the central dogma of what we call in molecular biology, how information is stored in the form of DNA, how it is translated into a form called RNA, and then is eventually, transcribed into RNA, and is eventually translated into the final product, which is a protein. Now, the common analogy we like to use here is parang nagluluto kayo. No, it's like cooking. You can think of DNA as your recipe in a cookbook, no? When you want to cook something up, you write down that specific recipe kasi alangan naman dadalhin yung buong libro doon sa kusina, baka mamaya masunog siya. So you write it down in the form of the RNA, and how you cook it will become your final product, or your protein in this case. And take note, not all dishes are the same. You can have the same recipes, but the final product will become different. And that is actually one large field in molecular biology in terms of how the proteins can have so many forms despite coming from the same recipe in the cookbook. But for now, we'll focus on DNA. No? And DNA is a very interesting molecule because it's very dense, and yet at the same time, it's very small. No? So the human genome in general actually has 3 billion, at least 3 billion base pairs. So the steps on the ladder, ganun kadami. And if you try to pull out all of those things straight in a straight line, it would actually be 1.8 meters in length. But the way your body stores it is that if you think about the cell, because the DNA is stored in the middle of the cell, the nucleus, if you think of that as the size of the Earth, a single base pair is like a person. That's how tiny these molecules are. So how should we go about studying them if they're that tiny? And this is where the concept of the PCR comes in. If you want to study something this tiny, you have to magnify it in some way. In biology, you use a microscope to optically enlarge things. But in molecular biology, in order to magnify the DNA, we utilize a technique that amplifies it, or pinapadami namin sila so that we can actually see it. And this is what we do when we actually run the polymerase chain reaction. And there's a machine down there. It looks like an ice machine. When I remember the first time I saw one, I thought it was an ice machine. But no, it's actually a very sophisticated machine. But saying that, now, the simplest way to look at it is it's like a photocopier. No? When we amplify DNA, what we're actually saying is we're making lots of copies. No? So, but instead of if a photocopier makes photocopies, no, a PCR replicates the DNA, and it uses the actual process in the body called DNA replication. So if you remember that image I showed you a while ago, where DNA is transcribed to RNA and is translated to protein, there's a little part down there, replication. So it's very important for the body whenever as it grows, that it continually replicates the DNA in order to preserve it. No, it's also some of the basis for how when you get your traits from your mother or father or things like that. But that's a different topic. But what the PCR does is it actually mimics this process of replication where the body makes copies of DNA. But in this case, we are taking advantage of this mechanism and making lots and lots and lots of copies of that DNA. So just like a photocopier, there are analogs, no? So this is what DNA replication actually looks like. You can think of it in terms of these components, no? If you have a big document na nakabind siya to a book, or maybe it has staples, you have to separate those pages. So you have a staple remover, no? You have the actual photocopier itself. You have a template, in this case, whatever you're trying to copy. You have a primer, which acts sort of like a marker to know what you want to copy. And then you have your ink, no? And in this case, you have, in the PCR reaction, it's your helicase, your polymerase, your template, your primers, and your nucleotides. Now we'll go over each of them. No? Your helicase, you can think of it as a staple remover. It acts like a zipper and it unzips the DNA. In PCRs, we actually use something different in order to separate the links. We use heat, no? So when you heat up DNA, it'll actually uncoil, and then it opens up and creates separated strands. This way, we can actually see the individual pairs that make up the sequences. And this process is known as denaturation or denaturation. So the first step, apply natin ng heat, no? Now, the next step, you need your photocopier. Now, the problem is, since we used heat to open up the DNA molecule, the problem is most polymerases, which is your photocopier in the biological system, doesn't work at that high temperature. No? So we actually use a polymerase that comes from bacteria that live in extremely hot conditions. In this picture, for example, you'll see the greenish orangey colors. That's actually algae. And this is around a geyser. No? So very hot ang water, sometimes beyond boiling point na yung water na to. But these cells are able to survive in these conditions because the polymerases inside of them can withstand such high temperatures. And that makes it ideal for use in our PCR. So we use these heat-resistant DNA polymerases so that they can operate as our photocopying machine while we heat up that DNA molecule. Now, the next, you need your template, and then you need your primer, no? So think of the source like your book. That's what we're trying to copy. And of course, you'd like to maybe copy a certain portion. You can think of it as bookmarkers. So you need to know where exactly you're going to start photocopying. In PCR especially, we're usually amplifying certain portions that we're interested in. You don't have to copy everything, no? So this is what makes it a very powerful technique, no? If you're thinking about studying for your exam, no, for example, in a textbook, you don't photocopy the entire book, diba? You only photocopy the section na kasama sa test. That's the same concept here. So, so you use primers in order to initiate the start of your replication. And then finally, you have your nucleotides. And you can think of this as the ink. Kasi syempre, you can't print anything kapag walang ink yung printer nyo. And these are the nucleotides. We call them the building blocks of DNA and RNA. And I mentioned it a while ago. So there's T and A and G and C, right? Now, RNA is a bit different. I won't go that much into it, but instead of T, it uses U. So it's slightly different, but the idea is essentially the same. So you have these building blocks to put it together. So when you put all of those factors together, these are the steps that happen. So you basically have your original DNA, you denature it to split it apart, and then you start sticking the primers to it, and then you extend your strands to create 2 copies. So that's how it works. Now, if you put those, what I showed you a while ago, it looks like this. So to denature, we apply heat. No, that's step 1. The DNA polymerase, you need that, your photocopiers to get into position. You start with your template strand and your DNA primers. So the template strand is your source, and your DNA primers are sort of like your markers to tell what section that you're going to copy. And then it'll start copying everything in the annealing step. And in order for that to happen, you need to have your ink, your nucleotides. And then once you're done, you have copies. Now this will continue on and on, the denaturing, the annealing, and the elongation until you have multiple copies. So you'll see in the first step here, there was 2 copies. And then from those 2 copies, another 2 copies, so naging 4. And then from there, 2 copies each, naging 8. And then from 8, naging 16. And you can see how this starts to snowball. Padami na ng padami yung mga copies of your DNA that you want, okay? So I have here a video so that you can visualize actually what it looks like, no, in a biological system. Where is the controls? Ayan. Sorry, ha? I have to find, where's YouTube? Ano ba to? Okay, there we go. All right. So there's your DNA molecule, no? So in order to start the polymerase chain reaction, we heat it up, and that unravels the DNA. There, you see it. Yeah. So there's that ladder, the step ladder. And eventually, when you hit a certain temperature, it'll split apart. So there, now you have 2 strands. Now we want to photocopy the strands. So is that a commercial? Okay, yeah. Okay. So there. Now the primers will stick to where they need. So you can think of that as the page marker going on the book, no? And then now we'll start the photocopying process. So your polymerase will attach to the area where your primers are. No? There's your polymerase. Okay. Nagbabuffer ba? Yeah. And then it starts copying using the nucleotides or the ink in the surroundings to replicate. And then it'll repeat itself over and over. So heat up, split, primers stick to the areas where you want to copy, polymerase goes to the area, starts printing, and then split again. So you'll see now we have 8 strands. No, the process repeats itself. Okay. And there. So by the end of the 3rd cycle, you already have your target. That's what you're trying to copy. No? And then so on and so forth. Now, after around usually 30 cycles, you would have made around 1 billion copies now. And that's exactly what we need when we are doing studies that involve analyzing your DNAs. So once you have enough, you can start visualizing it. No? One of the ways that we visualize the products when we're done multiplying it is that we run it through a gel. No? And the gel, I won't go too much into detail, but it allows us to separate it based on weight. And there's a very characteristic appearance of the bands depending on that weight. No? In the image here, for example, you can see that our sample number 5 doesn't have any bands. So that means the DNA that we were looking for wasn't present in the sample. And if you see 2, 3, and 4, they all have the sample. They're all the same line. So that means it's the same molecule, DNA target that we're looking for. But they have differences in the intensity, no? So number 2 is the most bright, and then it becomes 3, and then 4 is the least bright. So number 2 has more of the DNA that we were looking for compared to the other samples. So that's how you can actually visualize it. Okay. Now, there have been evolutions to the technique. One is quantitative. So a while ago, this is a medyo crude yung quantification method na to. But if you want a more accurate number of how many copies you've made, you can use what's called the quantitative PCR, no? We do it by including what are called fluorophores. So just to make it simple, we apply a light, a laser to the sample, and depending on the number of fluorophores it detects, it will release light at a certain intensity. So the way it works is there are these probes when elongation occurs, or meaning there's a target sequence that's being replicated, it'll hit the fluorophore and release it. So you can think of that as a dye. So the more copies you make, the more of this dye is released. And when you hit it with a laser, it will shine. And that intensity of the shining tells us how many copies were actually made. So the nickname for this is called the real-time PCR. Because as we run it, as more and more of the template is multiplied, we'll be able to see it rise as a curve. And the number of cycles or the number of times we'd had to run it, dictates the relative amount that there is. So the less cycles we take in order to hit a certain number of copies made, the more there is of that template that we're trying to replicate. So that's the idea of a real-time quantitative PCR. There's also the reverse transcription. So I mentioned RNA is a bit different. In order to make more RNA, we utilize something called reverse transcriptase, which allows us to convert RNA into what's called complementary DNA in order to use in the PCR. And this is important, especially when you're studying RNA, like for example, COVID or your SARS-CoV is an RNA virus. So we use Rt-Pcr in order to study the RNA. So in medicine, we use PCR in diagnostics. In cancer, for example, we can use it to detect certain mutations that are very important. You might have heard of BRCA in breast cancer, and also in ovarian cancer. So that will actually have, if you've heard of Angelina Jolie, she's one of the more popular examples of how this can be used as a sort of prognosis. If it's present in the family, there's a very high risk that it can be manifest in the form of those cancers. So her option when she tested positive for this was to have prophylactic mastectomy, or she had her breasts removed. You also have KRAS, in colon cancer. And this is also very important for us because it will dictate the kind of drugs that we can give. Because there are certain mutations in KRAS when present, we cannot give certain medications. So that's how we use it in cancer. And in infectious diseases, we use it in tuberculosis, for example. There is a test called the Gene Expert test that we use. It provides very fast diagnostic of the presence of TB, and at the same time also tells us if there's resistance to one of the first-line drugs called rifampicin. So it's very important in the field. And of course, more recently, we use it in the diagnosis of COVID-19 in samples that are sent to us. And I think this makes a good segue into the next part of our PGC talks, where I give it off to Dr. Uy, who will talk a little bit more about how PCR is used in the context of COVID-19. And with that, thank you very much.
Thank you so much for your very informative talk, Dr. Ayes. Before we proceed with our next talk with Dr. Uy, we would like to welcome our guest reactor, Mr. Alain C. Wayang. He's the section head of the Corazon Locsin Montelibano Memorial Regional Hospital Molecular Laboratory. Hi, Sir Alain. We thank you once again. Hello. Ayan.
Good morning.
Good morning. So, maganda pong na-invite namin kayo as na nagtatrabaho sa lab kung may additional insight po tayo na ma-share sa presentation ni Dr. Ayes. Kung pwede rin po kayo magtanong kay Dr. Ayes para po yung mga attendees natin ngayon magkaroon ng mas magandang idea kung ano ang ginagawa nyo sa lab dyan sa RT-PCR.
Actually, ma'am, yung presentation ni Dr. Ayes is already complete. So parang for my side, I don't have anything to ask him for clarification kasi nasagot niya yung mga nasa utak ko, especially yung the process of PCR from denaturation to the PCR itself. So I might ask this question, Doctor, kasi minsan marami tayong PCR test kits, correct po? Some test kits have 5 methods na inuulit, yung iba 4 lang. So is there a possibility na the other kit would be sold to a positive result while the other kit might give a negative result?
Okay, so if it's in the context of COVID-19, no, that's a bit different. But in general, a lot of these tests undergo validation studies, no, to in order to avoid yung mga ganong klaseng what we call them non-concordant results, no. But there is a small possibility that there can be some non-concordant, and usually it can be traced because there are what you'll notice about the PCR reaction, it can be sensitive to a lot of variables that may affect its results. No, as a molecular test, that's the reason why validation is important, not just in the kit, but even in the internal laboratory that's performing the test, because you need to make sure that there aren't any factors that are affecting the results. No, it's a bit beyond the scope of a high school discussion, but in the context of COVID naman, no. Because of the nature of the diagnostic itself, most of it is used in RUO or research use only situations. It is actually quite expected that if you're performing this test this many times, that there may be non-concordant results, which is why one of the reasons is when we report the findings is that it always has to be correlated to clinical findings and also yung related risk, or yung what we call the pre-test probability, the likelihood that the person is truly testing positive if he is positive. No. And just also to put into more context, usually PCRs are intended to be confirmatory. So for example, usually we have a screening test of some confirmed PCR. It's just that again, you're looking at the context of COVID-19, because there's so many cases and there's a need to facilitate the testing, we jump kaagad to PCR, no? So those are some of the factors. This is a bigger question of diagnostic use of PCR in mass testing diagnostics. But other than maybe some discordance between kits. But as far as yung, and sorry ha, medyo mahaba. The last one about one kit has that. This is usually based on the optimization protocol of that developer. So some of those differences might be small, like annealing temperatures are slightly different based on the prime. Those are some tweaks, kumbaga, that these manufacturers have developed based on their own validation studies and their protocols.
Thank you so much, Dr. Ayes.
Thank you so much for that very exhaustive answer, sir. So naliwanagan tayo na marami palang klaseng RT-PCR kits, no? Na hindi lang isa ang ginagamit natin. Dito po ba sa Philippine Genome Center, marami pong nag-donate ng RT-PCR kits? Ano po yung pinakaginamit natin dito na RT-PCR kit?
Yes, there are several kits that have been allocated and also donated to us. The one that we're currently using, of course, is a single probe BGI kit. No, it targets the ORF1AB, which is a very common target for a lot of the kits. We've also used Sansure. I'm sure a lot of people have heard of that particular kit, among others. So it's usually the choice of the kit is of course just you always have to test it with your internal protocols, no? So that's why even if we have a new kit, no, we have to make sure that it is compatible with the machines that we use, and you're able to to make sure valid yung mga lumalabas na results. So every time we get a new kit, we always have it validated.
Ganun po ba yun?
Mm-mm.
Sir, pareho po ba yung Rt-pcr testing sa yung ginagamit nyo sa pag-check ng variant? Iba yun, di ba?
No, they're different. Okay. So Rt-pcr uses polymerase chain reaction, no, the one I discussed. Sequencing, well, PCR can be a step in the sequencing. No? So when you get a sample, for example, you want to amplify it first, and then you create the library. We call it libraries from which we copy from. Because again, that's the power of PCR. It's one of the most powerful tools in molecular biology. A very small starting material, mas marami yung makukuha natin. You can think of it as, halimbawa, gumagawa ka ng project, syempre gagawa ka ng save file. So this is sort of like our backup kasi since very precious yung DNA samples, you really want to make sure that you have enough to work with because especially in sequencing, it's not cheap. the reagents we're using are not cheap. So you really want to make sure that when you run the test, something will come out. And amplifying the DNA beforehand gives us more room to work with para mas maraming template, DNA ang mapoprocess namin in a single run.
Okay. Maganda po to na si Sir Alain kapart ng discussion natin, no? Kasi yung setup niyo po dyan na nasa ospital po kayo, no? Mga ilan po ang ina-Rt-pcr niraran ng Rt-pcr every day?
Normally, ma'am, we run approximately for now kasi bumaba naman po yung census dito sa Bacolod for COVID. For now, we are running almost 150 tests every day. Last year po, umabot ang pinakamarami namin was 600 per day. Ganun siya kadami. However, dito kasi sa Negros, nung last year, the whole Negros Occidental na province is under sa kabilang hospital. Sa amin, Bacolod City lang. So, mas marami yung specimen na kinikater ng kabilang hospital, ng provincial hospital, kesa sa amin. As of now, kasi kakonti naman yung cases dito sa Bacolod, dun kami sa mga 150 specimens per day. But the capacity of the laboratory is up to 250 specimens per day.
That's a good information para malaman po ng mga nasa audience natin na ganun pala yung sa laboratory, no? Mag-answer pa po tayo ng questions mamaya sa Q&A. Maraming salamat Dr. Ayes and Sir Alain. Tatawagin ulit kita Sir Alain after ng talk ni Dr. Uy. But let me introduce our next speaker. She's also from the PGC Clinical Genomics Laboratory. She is our head pathologist. Siya ang taga-pirma actually ng results na nanggagaling ng Philippine Genome Center. She will talk about how do we interpret the Rt-pcr result. Let us all give a big round of applause to Dr. Justine Alessandra Uy. Dr. Uy, you may now share your screen.
Okay, so good morning, everyone. I hope you can see my screen clearly. So I am Dr. Justine Alessandra Uy, as mentioned by Ma'am Shalat, and I am the molecular pathologist of the Clinical Genomics Laboratory of the Philippine Genome Center. So my talk today, which is the second talk, is entitled Detected or Not Detected, Interpretation of the Rt-pcr Test Result. So I'll be discussing a little bit of an overlap from Dr. Ayes's talk, and then I'll also introduce the next talk that will be discussed during the next few weeks. Okay. So just a disclaimer, the images showing specific brands are mainly for demonstration purposes only and are meant to endorse a specific company or a specific product. So these are just examples. Okay, so for the outline, we have this. We will start with a review of the central dogma of molecular biology, then discuss the basic concepts of real-time reverse transcriptase PCR and interpretation, and then move forward with discussion of the selected SARS-CoV-2 Rt-pcr assays in the market, which are already actually discussed a while ago in the Q&A. And lastly, emphasize the importance of these RT-PCR assays in biosurveillance. So we'll start with the central dogma of molecular biology. Our understanding of health and disease continues to change with increasing detailed information about the human genome. So this central dogma can explain how our genes are actually expressed to proteins and are considered as the building blocks of life. So the aim of this part of the discussion is to simplify the concept. So pardon me if I don't go into the specific details, but it's just an overview. So at the microscopic level, genetic material is organized inside the nucleus of the cell, as pointed here. The transcriptionally active euchromatin are lighter in color, as you can see here, and the inactive portion are the darker ones, the heterochromatin. So although, as mentioned a while ago, that the genome contains around 3.2 billion DNA base pairs, only about 20,000 are protein encoding genes, so which are the blueprints for assembly of different structures in our body, as well as the enzymes and signaling molecules within the cells that make up our entire body. So the condensed form of your chromatin would be the chromosome. So for humans, there are 46 chromosomes, which contain the proteins and DNA that carry stored genetic information. So the first step of gene expression is called transcription, as previously mentioned, when the information of one strand of the DNA, so since it's double strand, there's just one template strand, is copied into a complement messenger RNA or mRNA. So the mRNA undergoes post-transcriptional modification. So for example, after transcription from DNA to RNA, there's splicing of the introns. So the non-coding regions will be removed, and then what's left will be the coding regions. And then a cap will be put on the 5' end, and the poly A tail will be put at the 3' end, and then it will be shuttled to the cytoplasm, where your translation will occur. So next in the cytoplasm, protein synthesis is the location. So it is translated from the language of nucleotides to that of the amino acid. So one or more mRNA nucleotide triplets. So for example, AUG or AAU, those triplets are called codons, and they will be equivalent to one amino acid. Or for example, if it's a start codon, then it's AUG, and then it will be equivalent to your methionine. And then there are 21 different amino acids that are used for protein synthesis. So the transport RNA is the one that would recognize the sequence of the mRNA, and then it's the one that attaches the, or is where the amino acid is attached, and then here as shown. So it was already mentioned, the concept of reverse transcription a while ago. So this is when the target mRNA is intended to be amplified. So in such cases, such as your RNA viruses, so for your HIV, your SARS-CoV-2, in this process, the mRNA is converted into your complementary DNA or your cDNA, and it is produced from mRNA, and it's used for, as mentioned, for RNA virus replication. So in terms of replication, actually what's most important here in the central dogma as it relates to your polymerase chain reaction would be DNA replication. So it is a semi-conservative process. So meaning each strand of parent molecule, so there are two strands, will be used to synthesize a daughter strand. So one, after it separates, one strand will also produce one daughter strand. So because the base sequence of the parent strand dictates the sequence of the daughter strand, replication is faithful, and the replication products would consist of, for example, from one double-stranded DNA, you can make here two strands, which will compose of two DNA strands, which will compose of one parent molecule and one daughter strand each. So as we've mentioned, this is the part that's important for polymerase chain reaction. So in relation to PCR, the assay permits replication of a targeted nucleic acid. So in this part of the central dogma is precisely the basis of our PCR. So let's move on to real-time reverse transcriptase PCR. So in order to understand this concept, what Rt-Pcr in real time is, we can divide it into three parts. So first, your PCR. This term simply implies that it's a chemical reaction that permits the synthesis of limitless quantities of a targeted nucleic acid sequence. And this is accomplished through the help of your DNA polymerase. So it mimics the replication process of DNA. Next is your reverse transcriptase. So as mentioned, this is the enzyme used to reverse the transcription from mRNA to your cDNA. So what happens is that your target mRNA will now be able to undergo um, replication through indirectly in PCR because your mRNA is made into the complementary DNA strand, and then it can be amplified. So lastly, real-time, as mentioned by Dr. Ayas a while ago, it refers to methods by which the target amplification and detection step occurs simultaneously in the same tube. So unlike other PCR techniques, there's no need for post-PCR processing. So as mentioned a while ago, this is your Rt-Pcr. Like any other PCR, it follows the same steps, so denaturation, annealing, and extension. So the specificity of real-time PCR can be increased by including hybridization probes in the reaction mixture, as seen in this photo. So this is actually the technique that's used for our SARS-CoV-2 RT-PCR. So for your probes, they are labeled with a combination of your fluorescent dye and your quenching dye. So one fluorescent dye serves as a reporter, and its emission spectrum is quenched by the second probe, so which is your quencher dye. So during extension, as you can see here, the nuclease degeneration of the hybridization probe releases the reporter dye, resulting in an increase in its peak fluorescent emission, resulting to the amplification as shown here in the plot. So for example, if we go back to the analogy of the book, or if you're using an ebook now, so it's sort of a highlighter that uses the find control F application. So it looks for the target DNA, and if the target DNA is present, then it will highlight everything and it will mark everything. So looking at the graph closely, here you can see the standard plot of your Rt-Pcr. So the baseline is the initial phase of your PCR. Usually it's from cycles 3 to 15 and contains possibly artifacts and background noise. So the threshold here in the dotted red line would be the cutoff separating your baseline and your exponential phase. So the cycle threshold, or CT, here is the PCR cycle at which the fluorescence passes the threshold, so which is set along the exponential phase. So here, you can see this is the exponential phase, and then the last phase here, which is your plateau, occurs once reagents are already depleted. So once we have a good understanding on the PCR plot, it's more or less easier to interpret a standard plot. So looking at this example, we set the threshold at the exponential phase. So this is the threshold. And then usually it's along the elbow of the curve. So the flat lines below the threshold are the samples with no amplification, or none of the sample or none of the targets are detected. So that's why it's called as not detected. So the increase in fluorescent emission indicates that the specific PCR product has been made. So that's why we label them as detected. And the intensity of the fluorescence is related to the amount of the product. So for example, the higher the concentration of the DNA, so the starting point is, if it's higher, then the fluorescence intensity at the end point will be higher. The higher the fluorescence intensity, the less the cycles are needed for detection. So that's why the CT is earlier. And then there's an inverse linear relationship between the log of the initial target concentration and the CT. So for example, looking at this curve again, so the first curve has a CT of almost a little bit over 19. So this is the first curve. And then the second curve here, 23. And then the third curve is 27. So we can assume that compared to the second and third curve, the first curve shows a higher DNA concentration. Okay, so interpretation goes beyond just looking at the plot. So it is important to follow certain quality control measures in order to ensure that the results are precise and accurate. So these are the following some of the quality control measures that are important. So first is to always inspect the Rt-Pcr plot. So for example, in this one, looking at this plot, the pointed curve that has the CT value of approximately 23, which in theory is considered as positive or detected, but since it doesn't show the usual, this is the usual sigmoidal curve, and then this is the curve of the pointed sample, it doesn't show the expected sigmoidal curve, then this is not considered as positive, and the PCR of this sample should be repeated. So next would be manually adjusting the threshold accordingly. So it's important to watch out for baseline noise or artifacts. For example, in this plot, we can see background noise here at the earlier cycles. So auto threshold from the uh, from the application um, that's seen here was set to include some of the background noise that passes through the threshold. So in this instance, the threshold should be adjusted upward so that all of the background noise will be under the threshold. So next, in order to check for validity of the run, the following controls should also be included. So positive control shows the ideal appearance of the amplification curve. And then it will also show if there are PCR inhibitors, because the positive control will won't show the expected curve that we want to see. So the negative control checks for contamination. So there are other negative controls, specifically your no template control, that ensures that the PCR reagents are not contaminated, and then the negative extraction control would ensure that the RNA extraction process is not contaminated. So this is another example of a run that shows amplification present. So this sample is actually supposedly the negative extraction control. But since this is observed, so this will lead us to uh, to the theory that there's a possible contamination that happened during extraction phase. So this will lead us to the next quality control measure, which is to repeat the run as indicated. So since we had a positive or amplified negative extraction control, then the whole batch under that control will be repeated or re-extracted. So it will depend on the protocol for repeat runs as indicated on the kit. And then as a result, the samples corresponding to certain batches or certain controls will be repeated. So it will also depend on the type of kit and how the kit was validated. So lastly, performance indicators may indirectly signal a possible systemic error or also signal a possible outbreak. So increased number of invalid runs may be secondary to incorrect swabbing technique. For example, the one who swabbed it is fairly new and still unskilled. So it can also reflect in the PCR runs, or if there's breach in transport protocol, and then all of the samples from one particular source will be all invalid, or there's a divergent from the standard PCR protocol. So another example would be monitoring for the positivity rates. So the positivity rates of all COVID-19 testing laboratories may be retrieved actually from the official DOH Encoding tracker. So this is the positivity rate from our laboratory in PGC. So as you can see here, you can see the daily samples tested here, and then you can also see the daily positivity rates, and then it's nice to compare them between the days or between that specific time period. And then for PGC, because our samples are heterogeneous, so meaning they come from different sources, it's also good to stratify these samples and then compare the positivity rate depending on the specific source. So in terms of reporting, it is important to be familiar with the parts of the test result. So first and foremost, the heading should show the laboratory letterhead as well as the contact details. Next, the body of the report must include the following. So specific to COVID actually is the addition of the patient's contact details. So tracking and contact tracing can be easier. And then the rest will pertain to the test performed along with test information and results. And also the remarks as mentioned by Dr. Ayas a while ago in the Q&A. So we always put the remark for clinical correlation. And lastly, the signatories for the medical technologists and as well as pathologists and laboratory staff that performed the test result. So this is an example of the Rtpcr test result from the Philippine Genome Center. So all elements that I've discussed are all present in our report. So before we move on to the discussion of the different SARS-CoV-2 Rt-pcr assays in the market, let's review the structure of the virus. So the virus has a single-strand positive sense RNA genome. So here with the following genes, you have your open reading frame or ORF, 1, A and B, and then your spike protein here, your spike gene that encodes your spike protein, envelope, nucleocapsid here, and membrane. So your spike protein facilitates attachment of the virus to your ACE2 receptors in the host cell. So they're usually found in the lungs, and then significant mutations involve this part of the genome because it primarily starts the attachment phase of the virus. So the N gene is at the end of the coronavirus genome as shown here. So this makes it the most abundant nucleotide sequence during virus replication. So it's a good target for PCR assays. The E gene has little sequence diversity between your SARS-CoV and SARS-CoV-2. So this is considered a more general marker for beta coronavirus. So if it's positive for E gene, it could either be 1 or 2. So the ORF1ab gene makes up most of the SARS-CoV-2 genome and encodes non-structural proteins, which are substances that are associated with viral replication. So the structural proteins are the ones that are shown here. So spike, nucleocapsid, membrane, and envelope. And then the other proteins are inside. So according to the WHO interim guidelines of diagnostic testing for SARS-CoV-2, suspected active SARS-CoV-2 infection should be tested with a nucleic acid test such as your Rt-Pcr and should target the SARS-CoV-2 genome. So there's no need to include actually the beta coronavirus target because SARS-CoV-1 or the original SARS-CoV is not in circulation. So for optimal diagnostics, at least two independent targets should be included. But for areas with widespread transmission, like in the Philippines, a single discriminatory target may be adopted. So such as the one that we are adopting now in the Philippine Genome Center. So PCR kits may vary based on a lot of things. So first would be the gene target. So we already saw the genes of your SARS-CoV-2 virus. So the targets may range from 1 to 4 depending on the design of the kit. And then for the CT cutoff, not all tests use the same CT cutoff value. So the range is from 35 to 42, depending on how they optimize their protocol. The cutoff is determined by test sensitivity and specificity. For the limit of detection, or defined as the lowest concentration of the analyte that can be reliably detected, that would also depend on how the kit was optimized. So the important note here is that the lowest concentration that is reliably detected. So not necessarily the lowest concentration. The kit can detect sometimes concentrations that are lower than the LOD, but if it's reliable, then but we're not sure if that number is reliable. So reporting parameters could also include stating that the gene is detected or positive, not detected, or negative. There are kits that show presumptive positive because they are positive for the beta coronavirus marker, the E gene only. So equivocal can also be seen in some of the kits, invalid depending on the kit used. So as mentioned a while ago, validation of samples should be performed to ensure the accuracy of the test. So all FDA-approved kits are actually qualitative. So therefore, reports will only include whether the viral genome targets are detected or not detected. So CT values are not supposed to be reported since its values are variable and are influenced by multiple factors. So for example, your sample, collection technique, transport condition, kit sensitivity, and many more. So here are some of the Rt-Pcr assays in the market. I have tabulated them according to their gene targets, limit of detection, cutoff value, and the results that we can expect from those. So there's actually a wide variety to choose from, and it will depend on several factors, such as, for example, your laboratory setup, how you optimize it, the staffing capacity. If it depends also on the protocol. If one protocol is more simple over the other, then you could choose that specific kit instead. So the last example here, which is the Expert Express, is a cartridge-based RT-PCR. So all of the steps from extraction and amplification and analysis are already included in one cartridge. So this is good actually for emergency cases because the turnaround time will be faster. But the drawback is that it's a one-to-one system. So one cartridge is to one patient. So in throughput is far less compared to your other RT-PCR assays. So the next few slides are just examples of the plots that we'll expect from the assays. So for example, this is the current kit that PGC uses. BGI is a single target detection assay. So take note of the presence of the amplification along the FAM probe. So this refers to your ORF1AB gene, and along the Cy5 probe. So this is the internal control, indicating that this run is valid. So the next one is from Sansure Biotech, and it targets two genes, so your ORF1AB and your N gene. And then this is another kit from Alplex, so which has three targets, so your RDRP, E, and N gene here. So aside from the kits that detect specific SARS-CoV-2 genome, several manufacturers also have products that can do multiplex testing along with other etiologic agents. So these are some of the examples that not only test for SARS-CoV-2 but also for flu and RSV and other etiologic agents. So some have already been approved by regulatory bodies, but some are for research use only status, such as this one, which is the Novaplex. So this is from Seegene, the same company as Alplex. They have an Rt-Pcr based assay to detect SARS-CoV-2 variants, which is still at the research phase. So as SARS-CoV-2 continues to acquire genetic changes over time, mismatches between primers and or probes and corresponding binding sites may reduce the sensitivity of our nucleic acid test. So that's why it's important to correlate them with biosurveillance. The WHO interim guidelines suggest that when using one target assay, there has to be a strategy in place to monitor for mutations that might affect the performance of the PCR assay. So that's what we're doing now. This table shows the SARS-CoV-2 variants from CDC. These are the variants of concern, which are said to be more transmissible compared to the original virus and might also escape neutralization of antibodies due to the significant mutations in the S protein genome. So for commercial assays, it is vital to keep track of possible incidents of suboptimal performance because of these variants. So in silico predictions of binding efficiency are initially done and suppliers release statements addressing the issue. However, in silico studies are insufficient to quantify the effect of a mismatch on the sensitivity of your nucleic acid test. So it's essential to also do experimental studies to compare the assay sensitivity for both variant and reference virus isolates. So as you may have already heard, there is a new variant that was detected in samples from the Central Visayas region primarily. So the way the variants are detected in the Philippines is that laboratories would submit specimens which were already confirmed as positive by RT-PCR and are subsequently sequenced. So most of these cases were confirmed using the Sansure Biotech PCR kit, which targets the N and ORF1AB gene. So we know that this particular kit can detect the new variant. So with the emergence of this variant, other kits, other manufacturers should follow suit and perform in silico and validation studies to check whether the kit performance is affected. So, as a segue, for more information on whole genome sequencing, stay tuned to the subsequent webinars from our other PGC colleagues. So, thank you very much for your attention and stay safe.
Thank you so much for that very informative presentation, Dr. Uy. And before we begin our Q&A, I would also like to call Sir Alain again, our reactor, if he has additional questions or insights, because he's a medical technologist and a section head of a molecular laboratory. So baka meron po kayong karagdagan na informasyon or mga tanong based sa experience nyo, Sir Alain?
Na naman, ma'am, so far, na-discuss rin ni Doctor yung lahat ng, even the kits was already been discussed. So, I think, I'm not sure if it's important, but it's not just the kits that we have so many kits to choose from. Also, for the extraction, we also have a lot of reaction test kits that we can choose from. So reactions or extraction kits may have two types. It could also be from magnetic na type of extraction or using the silica. Ang problema nga lang for us here as medical technologists, most na binibigay sa amin is the manual type. So pag marami yung specimen mo, parang down ka din kasi mas mahirap siya, mas tedious using the manual rather than have it automated. Tapos I would like also to answer, I'm not sure if our speaker can add to it. There is there's someone who asked about false positive. For us kasi yung false positive, normally makikita mo siya as late. Yung amplification niya is late. At the same time, most probable cause is contamination. Yun lang po.
So actually, I agree with Sir Alain, no? And in addition to that, if you're thinking or doubting of false positive result, then you can always repeat the run. So there are cases that you could repeat the run, and there are certain kits that would also trigger you to repeat the run. So for example, in Sansure, there's no need to repeat if there's only one gene that's positive. So if, for example, either of the ORF1AB or the N is positive, then you can report it as positive. And then, for example, for other kits like Daan gene, so they have the same gene targets, but what's indicated in the insert actually is to repeat the run if there's only a single positive target. So it would depend on how it was optimized.
Okay. So, ma'am, I guess it's time for us to be answering questions from Facebook. So, just a reminder for everybody, the Q&A box, I know Dr. Edsel has been very busy during the talk in answering your questions. But we do have questions from Facebook, and I would like the questions to be addressed to our panelists. First question, I'm also interested to know the answer to this. Okay, wait. What factors affect false positives? And is there a way to tell a result if it's false positive? So, ma'am, marami pong interested sa mga sa questions ng efficacy and false positives. Paano nalalaman kung false positive siya?
Well, in terms of looking at the result, if the result was already released, it went through a lot of different quality control measures to say that it's not a false positive. So if it was, for example, at the start of the analysis, we have a lot of the controls. So as mentioned, if nag-positive yung isa sa mga controls there, then possibly it would be a contamination. And then aside from that, the important thing is to look at the clinical picture of the patient. So it's a little bit difficult to say that, for example, an asymptomatic patient has a false positive result because that's the nature of the disease. More almost more than 70% of the ones who are infected with COVID-19 are actually asymptomatic or have mild symptoms. So before we jump into the conclusion that the report is actually a false positive, um, you have to know that before the result was released, a lot of things were taken into consideration before it was released as a positive result.
Okay. And ang context po nito, we're only talking about RT-PCR types of test, no? Hindi kasama dito yung ibang test na ginagawa for COVID-19 such as antigen, yung sa swab, yung
Yes.
saliva test, RT-PCR. So, yun yung, pero from what I remember, RT-PCR is pinaka-reliable in terms of parang confirmatory test. Tama po ba?
Mm-hmm. Because if you compare it with the other tests, so for example, if you compare antigen with RT-PCR, well, there are studies that would show that at a certain level it's concordant. But if, for example, an antigen test is negative and an RT-PCR test is positive, then you would lean towards trusting the RT-PCR test because it's the confirmatory test, and there's no amplification or there's no replication of the genomic material in the antigen test. So it's just whatever is the amount of or the concentration of the viral load that was obtained from the patient, and that's it. So that would lead to also a possible false negative. So that's why sometimes, if, for example, the patient is still symptomatic but had a negative antigen result, then we still proceed with RT-PCR as a confirmatory instead. But if it's positive, then most likely the the patient or the person is positive for SARS-CoV-2.
Okay, thank you for that, ma'am. Additional question from Facebook, from Mark Lawrence de Guzman. Do I always need a standard curve in each plate? And how do you prepare a standard dilution for Qpcr?
So for the standard curve, you don't need it for COVID because it's qualitative. The standard curves are usually intended for the quantitative assays.
Thank you for that answer, ma'am. By the way, we would like to shout out the students from Southeast Asian Institute of Technology from South Cotabato. Also, the Stratford International School from South Cotabato. Thank you. Thank you for the students in attending our webinar. We hope to see you in our next of the series. For all the STEM students who have taken the time to attend today, thank you very much for your support. We will have our next webinar on March 30, as mentioned by Dr. Uy. Do you have anything else to add, Dr. Uy, Dr. Ayes, or Aline?
I just noticed a question here from one of my old colleagues, Roland Remey. Hi. Siguro I'll just answer it. No, he was asking about LAMP. No, LAMP is a loop amplification. It utilizes a specific type of nucleic acid in order to perform it. So this falls under what we call the non-thermal amplification, because I mentioned a while ago in PCR we utilize heat in order to split the DNA molecule in order to do the PCR. But in LAMP, we actually don't need heat anymore. So we call those isothermal. Ibig sabihin, you don't need to change the heat. And this is very useful in situations where you do not have access to a thermal cycler. The PCR machine that I was referring to that looked like an ice box, That's called the thermal cycler because it cycles the temperatures usually from as low as 50 to as high as 92. But in isothermal setups, you don't need to change the temperature. You don't need to cycle it. So it's very useful in situations where you don't have a thermal cycler, like for example, if you're testing in the field. And usually what they do is you can just use a heated block element. Now, it's a very common tool that you find in the laboratories, and you can perform the whole PCR setup at a fixed temperature, usually around 40 to 50 degrees. So that's the great advantage of that type of PCR. There are some diagnostics. I won't advertise them, but they're utilized in the field, and it can be used for a lot of infectious diseases. Right now, there are some kits out there for malaria, for dengue, and right now, I think there are other kits that are also in development for these diseases. So thank you, Roland, for that question, and hi.
Yeah, in addition to that, we also have the TB lamp. And LAMP was also under development before for COVID-19, but I think it wasn't distributed throughout the entire world. I think one of the LAMP assays that was available for COVID was the one from Abbott, but it didn't reach us, I think.
Thank you so much for answering the questions. And if there are no more questions, at this point, we would like to thank all of the attendees for webinar series number 1, all about COVID-19 Rt-Pcr testing and its application, and talk number 2 by Dr. Uy, which is how to interpret the test results. Dr. Ayes, Dr. Uy, Sir Alain, thank you for joining us. Sa susunod ulit, kayo ulit pagsasalitain namin. Marami pa mga estudyante sa Pilipinas na kailangan marinig yung expertise nyo. And for the attendees, there is an evaluation form shared through our chat, and we will be keeping it open until 12 noon today. Those who have completed the evaluation form will be sent a certificate of attendance via the email address registered. If you have additional questions or if you are interested in the services of PGC, you can always email us. Don't forget to subscribe to our Facebook page and like our page. Our email address is our website is pgc.edu.ph. We also have a Twitter and an FB account at phgenome. Once again, this is your Ate Shilette, and thank you for attending, and we hope that you have a good day. Have a good week ahead. Bye, guys.